Dear student, today we will discuss translation. In molecular biology, translation means protein synthesis. So it is the molecular and biochemical process in which a protein is synthesized from the information contained in a molecule of mRNA in the form of codon. It occurs in cytoplasm of cell in both prokaryotes and eukaryotes. Let's discuss what are the steps of translation: preparatory step, translatory step, modification step. In the preparatory step, the activation of amino acid, tRNA and ribosomes occurs. In the translatory step, which is divided into three different sub-steps: initiation, elongation and termination. So protein synthesis occurs over here. Then comes the modification of whatever polypeptide is formed. So it comes under the modification. Now, what are the requirements of translation? The first important thing is ribosome. As we know, they are known as protein factories. Then amino acids. There are 20 fundamental amino acids which are required for the protein synthesis. In the protein synthesis, one amino acid is linked with another by the peptide linkage and form polypeptide, and these are known as building blocks of the protein. All kind of RNA are required: mRNA, tRNA and rRNA. Then several enzymes, because translation is a biochemical event, so enzymes are required to catalyze the reaction. Then here the energy sources that are used are ATP and GTP. Inorganic ions are required, such as magnesium, which act as a cofactor, as well as it is required for the assembling of two subunits of ribosome. Then various factors are required: initiation factors, elongation factors and termination factors. So these are the requirements of. Now we will start with the first activation of amino acid. Now activation means charging of amino acid with energy. The amino acids are present in cytoplasm in inactive state. In inactive state, they cannot participate in protein synthesis. So here the charging of amino acid occurs with ATP in the presence of enzyme amino acyl tRNA synthetase, and the out of ATP, the AMP linked to the amino acid, where pyrophosphate is set free, and this later on hydrolyzed to provide the energy, whereas amino acid link with AMP and enzyme to form a complex. This is called activated form of amino acid. Now this activated form of amino acid linked to tRNA. So let's look at the structure of tRNA and we will see activation of tRNA. So it has 3' and which is important, as the binding of amino acid occurs over here. This we have already discussed, and this is its anticodon arm. So I'll be representing this tRNA as this amino acid and its anticodon arm, because they are the main thing which participate in the protein synthesis. The tRNA is present in inactive state in cytoplasm, in which its 3' and is not attached to amino acid. The amino acid AMP enzyme complex formed above will be attached to 3' end of tRNA, and this results in the formation of amino acyl tRNA. So amino acyl tRNA is actually the activated form of tRNA. Okay, so same enzyme which is used in the activation of amino acyl tRNA synthetase is also used in the activation of tRNA. Now there are 20 different since there are 20 different amino acids, so there are 20 different tRNA. Okay, and they get attached to their cognate amino acid based on the anticodon, which is present in the anticodon arm of the tRNA. Now the tRNA charged with its cognate amino acid serves as the adapter, as it connects the amino acid to the codon on mRNA. So anticodon on tRNA bonds with the codon on mRNA. Now next step is the activation of ribosome. In the initial stage, the two subunit of ribosome get separated. The protein synthesis initiate on small subunit of ribosome, whereas the elongation occurs during the protein synthesis. The When the elongation occurs during the protein synthesis, the small and bigger subunit, they combine to form active ribosome. So for the initiation, only the small subunit is required, but for the elongation, both small and larger subunit are required. So the larger subunit of ribosome consists of two important sites for binding of mRNA and tRNA. So you can look at here. This is the small subunit represented as 30S in prokaryotes and 50 is the larger one. So larger subunit here, the two sites are clearly visible, peptidyl and amino acyl site. A stand for amino acyl site or acceptor site, P stand for peptidyl or donor site, and they combine and form the complete subunit, that is 70S. Okay, so this complete is required for elongation, small one is required for initiation. Now we will discuss the steps. So initiation is the first step in the translation of protein. The two subunit of ribosome get separated to the smaller subunit. The initiation factor IF1, that is initiation factor 1, initiation factor 2, initiation factor 3 bind along with GTP. The next is the initiator tRNA, that is formylmethionine tRNA. And now this is called initiator tRNA because it corresponds to the initiator codon on mRNA, which is AUG. Okay, so this tRNA carry the methionine. AUG actually codes for methionine, and this tRNA in the anticodon arm has a UCA as anticodon, and AUG codes for methionine, so it will carry methionine at its amino acid binding site. Okay, this is also known as formitable tRNA. Now the initiator tRNA bind to small subunit, and initiation factor 2 along with GTP help to identify the location of formylmethionine tRNA in P site of smaller subunit. Then the formylmethionine tRNA base pair with AUG on 5'-mRNA. mRNA has leader sequence called Sine-Delgarno sequence, which are situated upstream to initiation codon, and they bind to 16S RNA of 30S subunit, as the rRNA are present in the ribosomes. Okay, at 3' end of 16S RNA, this help to position the mRNA at P site. So small subunit along with mRNA along with formylmethionine tRNA along with initiation factor and GTP forms the pre-initiation complex. Okay, now this pre-initiation complex join to larger subunit to form the complete ribosome. When the two subunit of ribosome bind, the GTP is hydrolyzed to provide the energy. So till here 1 GTP is used up and 1 ATP, which was used up in the activation of amino acid. So 1 GTP and 1 ATP are used up. Now you can look at the structure here. So this represents formylmethionine tRNA, the first tRNA, and it joined to AUG on mRNA with the anticodon arm. Okay. Now next step is elongation. So elongation consists of 3 different steps. Binding of amino acid tRNA. This is second tRNA. First tRNA was initiator tRNA. The second tRNA was the which correspond to the the next codon on mRNA. Okay, so the binding of amino acyl tRNA at you can say A site, formation of peptide bond and translocation. So next we will continue in part 2. Thank you.